We do have a lot to cover today, so let's start with the C double bond T happens. As you can imagine, that's a misspelling on the DNA bonding. So basically, any mutation or any change in the DNA sequence, in the genetic material, that is defined as a DNA mutation. But it's very important to keep in mind that DNA mutation is not referring to what normally happens during crossover. Now, you know, during crossover meiosis, you have interchange of genetic information. You have those genes that would cross over to the other side, right, to the other allele. Now, when you're talking about DNA mutation, you're not talking about that. That is everything else that happened but recombination events. Now, we're going to cover several types of mutations. We have to cover spontaneous mutation, and also we have seen a lot of environmentally induced mutations. We're going to cover free radical mutation, a little bit about the drug mutations, so you can have an idea of drugs that are very deleterious to DNA sequence, and some of the forms of spontaneous and induced mutations. What is it that mutation is really implying, or what is it that is doing? We know that evolution would not be possible without a mutation. And then we have covered before another very important thing. Usually mutations would happen between genes, I would say, contrary to crossing over and recombination that only happens among allele chromosomes. Mutation can happen among genes that are not alleles. That usually happens between genes that are not alleles. Now, this is just an idea of how is it that mutation can be beneficial. Yes, we covered this about, you know, we have a slightly different protein that has a gain of function, so the organism ends up with a better system for processing of proteins. Now, we had covered this, which is an inversion, DNA inversion. If we have a piece of DNA, best way to explain it, just draw it out. You have a certain sequence, and if there are cuts on this particular DNA, 2 cuts, and it happens that DNA flips around and it connects again, so now you have an inversion on the DNA sequence. That's an inversion mutation. So these are mutations that happen on a big scale. These are the mutations on the chromosome. Remember, I had mentioned 2 forms of mutations before. There's the small mutations and the big mutations. And the way that they were classified as small and big was that the big mutations could be seen. They were visible changes. Okay, so what is a small mutation? A small mutation, you can have a specific base that was mutated into something else. Like, for example, an A that gets mutated to a C or to any other of the bases. So that's in a very small segment scale. So when you cannot really see with the microscope, or you cannot tag it and see with the chromosomes, those are the small DNA mutations. Now, when you have big mutations, those are the chromosomal mutations. And so we have the inversion, that is one of them. Now, how does that happen? We had covered this last week also, that if it happens that the DNA, you know, DNA is always coiled around and packed into those histones. So if it happens that a couple of nucleases reach the DNA and cut it on 2 sides, and you know the DNA is all coiled up like a rope. If it happens that DNA has to ligate again and it does not ligate the proper way, ligates on the opposite way, so this causes an inversion of that segment of the chromosome. Now, you have several types of inversions.
C is for clue, that's okay with me.
That you can detect based on what happened to the chromosome. Does it involve the centromere or not? So here you see one inversion that is called paracentric, that occurs in only one side or in one arm of this chromosome, and it does not involve the centromere. So when you have paracentric mutations, both arms retain their original size. Now, if you have a pericentric mutation, that means that the centromere is involved here. So when that happens, usually you have a change in the length of the arms of this chromosome. Now, as I mentioned here, inversions can be a problem for the gamete formation. Problem would be here that the chromosomes will not recognize each other, and the homologous chromosomes would have just a hard time finding their respective pairs. So basically, a lot of times it's going to be embryonically lethal, but it can cause a lot of developmental problems, or loss of function or gain of function problems. Now, another type of big gene mutation is translocation. So you can have two types of translocation. One type is that one chromosome will donate one piece to another chromosome. So this is called non-reciprocal translocation. Now also you have the reciprocal type, which I don't have the picture there, but you can imagine the reciprocal type. You have a piece of this chromosome here that also gets cut. So now this chromosome, it donates as it gets. Now non-reciprocal, one gets it all. Reciprocal, this one will also lose a piece and donate to the other one. Now one example of a syndrome that is caused by this type of mutation, I don't know if you have heard about. Anybody can tell me? For the non-reciprocal translocation. Doesn't ring a bell? Okay, I'll give you a clue. It's a syndrome that we just seen last week, and is also caused by non-disjunction of a small chromosome. No, that's loss of a piece. Loss of a piece. Down syndrome, yes. Down syndrome. Now, how can that cause Down syndrome? Any idea? Yes, it will mimic the trisomy of chromosome 21. If this here is chromosome 21 and it loses a big chunk to another chromosome, it so happens that chromosome 21 will pass most of its genes to that other accepting chromosome. And the child will be born with symptoms of Down syndrome, everything, all the phenotypes of Down syndrome. But when you do the karyotype, this person does not have trisomy 21. And then you look at the other chromosomes, and if you do tagging or if you do chromosome mapping, you will see that one of the other autosomal chromosomes has received most of the genes from one of the chromosome 21s, from one of the parents. When you have a reciprocal change, usually involves generation of cancers. And I'm going into more detail for that. Okay, so how is it that translocations can happen? When you have your two chromosomes that get close together, non-homologous, during regular cell division, you have cuts that can happen or in the two sides, and then if they don't join the right way, they can just translocate pieces of DNA. Now with translocations, it's important to notice that you do not lose any genetic information, because one piece is just shifted to another side, to another chromosome. Now, you can find it if you do the karyotype and genetic mapping, chromosome mapping, you can find where that piece ended up. Now, when is it that these translocations create problems? They create the problem with genes, for example, that are usually transcribed and expressed, like housekeeping genes. Those are the genes that you have to have transcribed all the time, like as you remember from lecture 6. If those genes are translocated to another chromosome and that translocation happens near the centromere, that is a problem, because near the centromere is a place that very low transcription will take place. So close to the centromere means not a lot of transcription happening over there. So if you shift a very important gene, of housekeeping gene for the cell, you shift to a place close to a centromere, the gene is going to slow down the expression. That means the cell is probably going to die, or is going to have a lot of problems. Now, the same can happen on the other way. You can have a gene that should not be constitutively expressed, and if it happens to be mutated and to shift to a place in the chromosome where you have genes that are constitutively highly expressed, so now you have a problem. or for example, as they say here, if you have a gene that is expressing some death signals, and it gets to be constitutively expressed, so the cell will just commit suicide. It's going to keep on telling the rest of the cell or other neighboring cells to just come and kill me. And it was not supposed to do that. It was just a problem with the translocation of that particular gene. So that would be the case of a control of a gene due to a position in a different promoter. You know that what is it that is controlling the gene expression? It's the something that, yeah, so it's a promoter. So if you put a promoter in a gene that is not supposed to be there, so now you're not going to have the proper expression of that particular protein. Now, as I mentioned here, translocation can lead to cancer, and then we're going to study a lot more of this in the next lecture when there's a cancer lecture.